1, 2, 3, action.
We have glycolysis and alcohol fermentation and lactate fermentation. Glycolysis takes place within the cytoplasm outside the mitochondria and is the breakdown of 6 carbon to 2 carbon pyruvate molecules. Fermentation, an anaerobic process that processes a limited amount of ATP in the absence of oxygen. Alcohol fermentation is where pyruvate is converted to ethanol in 2 steps. First step, release of carbon dioxide from the pyruvate, which is converted to the 2-carbon compound acetaldehyde. Step 2, acetaldehyde is reduced to NADH to alcohol. Lactic acid fermentation. Pyruvate is released directly by NADH to form lactic acid as an end product with no release of CO2. This happens in animal cells and it happens during strenuous activity when one's muscles need it immediately. For glucose or glycolysis, you start with your glucose molecule, you get some ATP to ADP. It has to have the 2 ATP to get her going. Then it splits and produces 2 3-carbon molecules there and there. Then it goes to this, and the NAD to NADH. You add a phosphorus in there to get and oxidation of the G3P occurs as NAD plus receives high energy electrons. And then it goes to BPG, where ADP turns into ATP. And becomes that. See, phosphorus is gone on that side now. That's a substrate level ATP synthesis. And then it goes down to the 3PG, which is the oxidation of that occurs by removal of water. There goes your H2O, and you've got all this fun stuff here. And it goes to the PEP, which is another substrate level ATP synthesis, and that goes down. And you get your 2 pyruvates right there. And you get a net of 2 ATP because you start with 2, you add 2, boom, you get 2. Fun fact.